Click for IB Biology. Here we're taking a look at an overview of the synthesis of proteins. There's a blank PDF of this presentation down in the description. You can annotate that with your own notes. There's a quiz, 10 questions with answers, and the channel link has got a link to an AI prompt. You can use that to build your own self-assessment, individualized assessment of this content. So, protein synthesis in eukaryotic cells and prokaryotic cells has a number of differences. Of course, with eukaryotic cells, the process begins with the DNA, the chromosomes, inside the nucleus. Whereas in the prokaryotic cells, the DNA is the looping structure found within the cytoplasm, like so. Eukaryotic cells, the section of the chromosome called the gene is copied into a form called messenger RNA. The details we'll look at in the next videos. That's called transcription. So we copy the gene into messenger RNA. And the messenger RNA in a eukaryotic cell has two possible destinations. One can be what we call free ribosomes. They're unattached in the cytoplasm, and they'll build the protein on those free ribosomes, and we know that this means that the protein will be used inside the cell. It will be an intracellular use of a protein, so it could be an enzyme within the cytoplasm. On the other hand, the messenger RNA might locate itself on the ribosomes on the endoplasmic reticulum, in which case, when translated into the protein that they're using is going to be used outside of the cell, and so there'll be some kind of secretion process taking place where that protein is released from the cell, such as a hormone. Down in the prokaryotic cells, we don't have the nucleus, the endoplasmic reticulum, just free ribosomes, and you'll recall that these ribosomes are smaller than the ones in eukaryotic cells. They're given the 70S size, whereas these ones up here, these are the 80S size Svalberg units, that's called. So one or two differences there. Let's look at the diagram. This is a diagram of protein synthesis. I'm trying to draw attention to the stages of protein synthesis. And the first stage is where the gene is copied and rewritten in the form of messenger RNA. And I was showing that in this here. And of course, in a eukaryotic cell, this takes place inside the nucleus. The messenger RNA here is shown not to scale. I'm just making it larger for the purposes of the diagram. In fact, it would be roughly the same length as the gene itself. So the messenger RNA then leaves the nucleus from the transcription. Transcription occurs first, and then the messenger RNA will locate itself out into the cytoplasm on the ribosomes, which themselves are composed of RNA. And in fact, in two units, a small subunit, which attaches itself to the messenger RNA, and then the large subunit will be responsible for bringing in the amino acids. Now, those amino acids are circulating in the bloodstream. They're derived from our diet. We've eaten protein, digested it to its amino acids, absorbed those into the blood, and then they're taken into cells, and they are locked onto particular tRNA molecules. So you'll recall that there are 20 different types of amino acid, and each type of amino acid has its own type of tRNA molecule. So very specific. That attachment process is called activation. Now, with the messenger RNA located here on the ribosome, a mechanism exists which allows us to bring in the correct amino acids at the correct time in the correct sequence. It's called codon-anticodon binding, and we'll study that in our videos on translation. So now we're into the heart of the translation process. So we're building the amino acid sequence, the polypeptide. And it also turns out that, interestingly, the messenger RNA usually has more than one ribosome located upon it. And so we can see here that this polypeptide is in an advanced stage of building. This one's slightly behind it and just beginning here. And so we have multiple ribosomes on the same messenger RNA, and this is called a polysome. Or you might like to think of it as a polyribosome. Okay, so let's just summarize again. Transcription is turning the DNA into messenger RNA. And then translation is turning the messenger RNA sequence into the polypeptide sequence. So we go DNA into messenger RNA is transcription, and then messenger RNA into the polypeptide, that's translation. A link to a video on transcription details is now occurring here. We've got the link to the channel and, of course, to the playlist for this entire topic.